it's a direct, you know what, that's another thing, is if you send me a direct message, everyone is going to eventually see it, right? Because it's screen record. Somebody sent me a direct message saying that we're going to be famous on YouTube. But you guys will see it. Okay, anyways, okay, we got a focus mission. All right, so what is standard deviation? Like I said, we don't need to memorize this equation. It's basically a formula that calculates just how all the little data points, all the data, how varied it is, okay, how varied it is. And so what is it though? If we break it down, and again, you don't need to memorize it, it's going to be given to you, but what is it? So S is the symbol for standard deviation. It's a square root of the sum. It's a sum of all the data points minus the mean of all the data points squared divided by the sample size. Okay, so that is standard deviation, and once standard deviation is calculated, then on a graph it can be showed. Okay, so let me move here. Okay, so if you use that equation and found all of, you calculated all of standard deviation, and you were to calculate it, you would get 0.73. Okay, and this is the mean. So on a bar graph, why does it keep on saying this? I'm sorry, you guys. Something's popping up on my screen. Oh no, please don't tell me. What happened? Hold on. Oh no, and I'm screen recording my password. No bueno. This totally sucks. This... I don't want to screen...
2 hours later.
Yes, you guys are biologically smart too. Okay, so standard deviation is calculated. Then what do we do with it? If you were to graph this, and you won't be asked to graph it, but you might see it on a graph. Like I said, for the AP exam this year, you guys are going to have to analyze a graph. So you might see these bars. Okay, so you'll see a bar graph. The top of the bar graph is the mean. Okay, so the top of the bar graph is, that point right there is 98.25, but then the plus or minus would be the standard deviation, so plus 0.73 minus 0.73. Okay, so that's the top of the bar, and then this is the bottom of that standard deviation bar. Okay, so this is showing you visually that the top of the bar graph bar is the average or the mean, whereas above and below, that's the variance, the average variance, okay? All right, so that's standard deviation. But then, now we have to talk about what's actually more important. Is the 0.73 given in the problem? Yes, so that's a good question. If you were to calculate it, and I don't think that they'll ask you to calculate standard deviation, because look at all these data points. That would take too long. But most likely they would give it to you, and then you might have to plug it in to the standard error of the mean equation. And this is the equation. Again, all of these equations will be given to you. The standard error of the mean, this is what we actually use in AP biology, okay? Standard error of the mean, the equation, so standard error of the mean. I know it kind of looks like it says sex on there, okay, but it doesn't say sex. It means standard error of the mean, okay? That's the symbol for mean, okay? Is equal to standard deviation divided by the square root of the sample size, okay? So what is standard error of the mean? It allows you to infer how well the sample mean matches up to the true population mean. So like, for example, if I wanted to find out the mean or average age of students at our school, I could take a sample. I could take like 50 students, and then I would find the average age of those 50 students. But then, is it truly representative of the entire population of our school? Then you could do a standard error of the mean calculation, which could give you a better idea. Okay, so it's going to help you determine the confidence in the data collected in the sample. So in AP Bio, we're going to use 95% confidence interval, which is plus or minus 2 SE, standard error of the mean. It's 2 times this. It's 2 times this, okay? Standard error of the mean. Random sampling of the population should produce a mean that falls within plus or minus 2 SE, which is 95%, which means that 95% of the time when you take a sample, it's going to fall within that range. Okay, so let's actually try to apply this. I think it'll make more sense if we just continue on and you'll see examples of standard error of the mean. Somebody asked, can it be any sample size or is there requirement to the minimum sample size? So, you know, with science experiments, the larger sample size, the better. So, if you were asked to design an experiment, try and go with like a large number sample size. Depending on like what it is, like if you're doing an experiment with mice, maybe you can choose 100 mice. If you're dealing with humans, maybe you can say like 1,000, 1,000 human sample size. Okay. So, anyways, if you were to calculate oh wait, I gotta let somebody in. So, if you were to calculate the standard error of the mean, okay, then you would get 0.06. Oh my gosh, sorry. I was trying to grab my annotation tool. Okay. So you would get 0.06. Does everybody understand how we got that? It is standard error is equal to the standard deviation divided by the square root of the sample size. That's how we got 0.06. Okay? Hold on one second. I gotta let more people in. So standard error is 0.06. All right? What do we do with that? Well, we need to multiply it by 2. Okay, we need to multiply 2. Again, in AP Bio, we have to multiply the standard error by 2. Okay, so 2 times 0.06. Somebody asked me, wait, the square root of what? Oh, n is sample size. Remember, sample size is n or big N? That is the symbol for sample size. So going back to this example of body temperature, the sample size, I believe, was 130 people. We took the body temperature of 130 people. So it would be square root of 130. Okay? So that's how we got standard error, was you got the standard deviation, you divide it by the square root of 130. Okay, but what is... I gotta let somebody else in. So what is 2 times 0.06? It's going to be 0.12. Okay, so, wow, my pen is now really messing up. So 2 SE X or SEM, sometimes you can see it as, wow, what is up with my pen? SEM. Okay, so I've seen both on the exam before, SEX and 2SEX or 2SEM. I've seen both. Okay, so you're going to graph that. On the exam, you will see 2SEM or 2SEX. Okay, so this is that bar graph again. Remember, the mean is 98.25. But then when you add the 2SEX or 2SEM, it's going to be 0.12 above, 0.12 below. Okay? So notice how the bar is a lot shorter than standard deviation. Do you remember standard deviation was a lot bigger? So what does the 2SEM mean? It means that it tells you how close your sample mean comes to the true mean of the population. 2 times SEM gives you a wider range, giving you 95% confidence that a sample mean and the true mean will fall within that 2 SEM range. So before you multiplied it by 2, it was, it used to be 0.06, okay, 0.06. And that is, above and below the mean. That's a very narrow, it would be a lot shorter. The bar would be a lot shorter. So multiplying it by 2 gives you greater confidence that the true mean is going to be somewhere within this range, okay? So that means the 2SEM means that you're 95% confident. You guys, this pen is just, I don't know what's going. Oh, you know what? Maybe it's because I'm screen recording and it's like making my computer not perform very well, okay? But that range right there means that you're 95% confident that the true mean of body temperature of the entire population falls somewhere there, okay? So on the AP exam, you guys are definitely going to see 2SEM, or I would have told my students, oh, be ready to graph 2SEM, okay? So this is standard deviation compared to 2SEM. Standard deviation shows you an average deviation from the mean. See how the bar is like really, there's a large range, okay? Right here, it's a large range. But when you look at 2SEM or 2SE, it shows you with 95% confidence that the true mean or sample mean will fall within that little range, okay? Why use 2? That's a good question. Why 2? Because 1 SE is too narrow. It's too narrow. Remember, 1 SE was 0.06 plus or minus 0.06. But if you multiply it by 2, oh my goodness, my pen. If you multiply, that's supposed to be a 0.06. But if you multiply it by 2, it's going to be 0.12. So that is going to give you a wider range, and therefore you are 95% confident that it's going to fall within that range, okay? I think if it's 1 SC, guys, I don't remember, but I think if it's 1 SC, you're 60%. I think it's 60% confidence that the true mean falls within there, okay? Somebody asks, Pecan asks, you said we probably won't have to calculate standard deviation. So if we got a standard error problem, would they give us S? Yes, definitely. Definitely. Okay. Oh, okay. Somebody watched the Edpuzzle. Okay, thank you so much for that clarification. So 1 SE is 68% confidence, but with 2 SE, you're 95% confident, okay? Thank you so much for that clarification. So what does this tell us about the data set, okay? Hold on, let me just scroll up. Okay, no, it doesn't. What does this tell about the data set? Remember that table with all of those body temperatures? The mean, after calculating the mean, it was 98.25. The standard error was 0.06. But then for AP biology or for biology or biology in general, we're going to multiply the standard error, that's the SC, we're going to multiply it by 2, and that gives us 98.25, which is the mean, plus or minus 0.12 degrees Fahrenheit. So when you add 0.12 to 98.25, that gives you a range. It goes all the way up to 98.37, and then when you subtract, sorry you guys, when you subtract 0.12 from 98.25, that gives you, it brings you down to 98.13. So that's the range. Do you guys see that? That's the range. We're 95% confident that the true body temperature mean of the population is going to fall within this range, 98.13 to 98.37, because it's plus or minus 0.12 from that mean that was calculated, okay? So the range for the sample mean, actually, so now we're just doing the analysis part right here, it does not include the commonly accepted population mean body temperature of 98.6. Do you guys see that? Isn't it below? I mean, we always say, oh yeah, human body temperature is 98.6 Fahrenheit, right? 98.6 is not in this range. So this suggests that there is something going on with the group of people that we chose. This suggests that there is a statistical significant difference between the mean of this sample and the mean of the entire population. So something was up with, and let me scroll back up. Yeah, something was up with this group of 130 people that we chose from our sample. Their body temperature is somehow, their range is lower. Their range is lower than the commonly accepted temperature of 98 point, I'm sorry, not lower. Yeah, it is lower, sorry, it is lower. 98.6, okay? They have fevers. Oh, reverse fevers, right? Their temperature is, for some reason, just lower. But let's just try to apply this to an actual practice problem, okay? So let's see here. A student noticed that the ivy leaves growing on the shady side of a building were larger than ivy leaves growing on the sunny side of the same building. The student collected and measured the maximum width in centimeters of 30 leaves from each habitat. Use statistical analysis to determine if it's likely that there is a significant difference in leaf size between the shady and the sunny ivy leaves with 95% confidence, plus or minus 2 SE. So you guys, I have seen questions like this on the AP exam before. You do not have to graph because, again, you guys are taking the digital version, so you will be given a graph, okay? So this is the collected data, so you would be given a table. So for shady leaves, the mean leaf size was 7 in the shady side of the building. In the shady side of the building, the mean leaf size was 7.43, whereas in the sunny, it was 5.88. So there's definitely a difference there, right? Standard deviation was calculated. The sample size was 30, so it is equal. And you know what? In an experiment, you do need to make sure that all experimental groups and control group, which there is not a control group here, but make sure that the experimental groups, they have equal sample size, okay? And then the standard error was calculated as well. So if you were to graph this data, what would you guys be graphing? You would be graphing the mean, okay? So like you would have like 2 bars, okay? This would be like 7.43, this would be like 5.88, and then you would have to graph also the SE, okay, the standard error. But in AP Bio, are we just graphing the SE? What do we have to do with the SE? We have to, remember, it's, yeah, we have to multiply it by 2. It's plus or minus 2 SE. Okay, so we have to multiply this by 2. Somebody asked me a question. Would they give us a table already filled out on the AP exam? Oh, no, yeah, no, you would, okay, you would be given a table. They wouldn't ask you to find that out for yourself. They would give you a table, okay? So let's move on to the next slide. So I would have you, so this is actually where you guys, like I said to you guys in class, this Google Doc, there's not a lot of writing, but I do want you guys to write here, okay? So on your Google Doc, if you have it up now, you can do this right now. What is the 2SE 95% confidence for shady and sunny set data? So then you would just multiply by 2. So over here you would type in 0.60, and then over here you would type in 0.48. Okay? All right, you would do that. You could take a picture of it if you want. Okay, and then, okay, somebody asked a question. I'm still a little confused. What is the difference between standard deviation and standard error? Okay, good question. So, standard deviation, it basically shows the variance of the data. So it shows you just how the data varies. Like you see here, there's 130. There's like 130 data points. 98, 96.3, 97.8, 98.4. I mean, there's 130. But standard deviation would show you, like here, this is standard deviation. It shows you the varying data points in that sample set, okay? Whereas standard error, it's basically telling you where the true mean is predicted. The true mean of that data set is probably going to end up. It's going to be somewhere within this bar, So standard deviation, it's only telling you about the data set, whereas 2SE or standard error is trying to tell you where or how far they think, 95% confidence level, we think that the true mean is going to fall. They're basically measuring two different things. They're showing two different things. One shows you the variance of the data. The other one is saying, oh, we're 95% confident that the true mean is somewhere in this little, you know, the bar. I hope that makes sense. Somebody says, Sofa says, so the only thing we need to calculate for this section of the AP exam is the 2SE. Yes, okay. And then somebody asked me, wait, can you explain how the confidence is established and used? That is a good question for a statistician, okay? Why do they use 95%? I don't know. You can ask Ms. Martin, who is the AP Stats teacher. I am sorry, but I just know that in AP Bio and in Sciences, they always use 2SE, which is a 95% confidence level. They've just picked it, and I don't know exactly why, okay? Oh, interesting. Somebody said... And you guys can't read this until you see the screen share. Somebody said, so SD, standard deviation, stands for super diverse. Get it? Super diverse because it shows you the variance. And SE, standard error, means super expected. Oh, that is so good. Like, it's expected to be in that range. Ooh, so super diverse shows you the diversity variance. And then super expected is where we think the true mean falls. That is so good. So that was Tony Stark that said that, and I don't know who that is, unfortunately, but that was Tony Stark. So Tony Stark is a genius, huh? Okay, going back to here. So what would you do next is you would graph the mean, like I said this, okay? I already said this, right? You would graph the mean. You would have two bars, right? And then the top of the bars would be the mean, but then you're going to add the two SEMs. The two SEMs, which are these, plus or minus, plus, minus, plus, minus. All of a sudden, my pen is working great, okay? So somebody asked me, how are you going to do that on the exam? You can't. Because you're taking the digital version, so instead you'll be given a graph, and then you'll have to, I don't know what kind of questions they'll ask you. Oh, there's practice ones that I'll show you where you're going to have to apply your knowledge of what 2SEM means, okay? So you're not going to be graphing, but you're going to have to use that knowledge of the 2SEM bars and what they mean to the exam. You have to use a calculator on the exam. So I want you guys to have a calculator ready when you take the exam. Okay? Okay. So I want you guys to do this on your own. Let me show you how to do this though. I want you to graph the mean. So for shady it's 7.43. So this is how you do it. You can insert a shape. Like insert a rectangle shape. So shady is red, right? Which was 7.43. So maybe you can do something like this. And I'm not being very accurate right now. But so this is how you would do a bar. And then you see this icon for fill color. You would go to fill color and choose red. Okay? And then you know what I find to be really easy in Google Slides or Docs is just to copy and paste. So I'm going to copy Control C or Command C and Command V, and I'm copying and pasting this for Sunny, but I'm going to change the color to blue. So this is what you need to do on your Google Doc on your own time, okay? And Sunny was, what was Sunny? It was 5.88. Okay, 5.8. So I'm gonna lower this to 5.88. 5.88. So you're gonna do something like that. What did you write for slide 25? Oh, for slide 25, I just multiplied 0.6 by 0.3 by 2. So you're gonna write, 0.6, and then here you multiply it by 2, 0.48. That's what I wrote for slide 25, okay? All right, so going back to the graph, even for 24, yeah, you know 2SE? 2SE is 2 times the standard error, so it's 2 times 0.3, 2 times 0.24. That's how I got these numbers, okay? Yes, I will be recording this and I'll put it up tonight, okay? Okay. Oh, okay. Here we go. So, and then add the 2 SE bars. So you guys, the 2 SE bars is plus or minus 0.6. Is this test for tomorrow? Oh wait, sorry. Somebody asked me, do we have to write plus or minus on the slide for 25? You can. I think that would be actually a very good idea to write plus or minus 0.6, plus minus 0.48. That would be a good idea. Okay. And then is the test for this tomorrow? The test tomorrow will be based on, it's 10 multiple choice questions, and 10 multiple choice questions, and there are sample multiple choice questions in this Google Doc that let's get to those, because it's all about like analyzing the data and then choosing the best answer. That's what the quiz is going to be on. So it won't necessarily be about graphing, but it's about analyzing the graphs. Okay? I know, I'm so sorry, Sofa. And student. So I want you guys to add the bars on your own. I'm sorry, guys. So let's go ahead and 0.6 above. So you're basically going to be adding 0.6 above, 0.6 below. Okay? And we don't have time for that. But let's try to get to... you know what? I think that we do need to draw the graphs. We do need to draw the... So 0.6... hold on. 0.6 above... Why do I feel like I'm not getting a line? Okay, there we go. 0.6... and just kind of estimate 0.6 above, 0.6 below. So there's my line for 0.6, and then let's make it a little bit thicker so I can actually see it. And then the other one is 0.48 above and 0.4 below. I think that's like about 0.4, like that, okay? Oh wait, I need to make it a little bit... Why is it not elongating? That's super long. That's probably too long. Okay, but so you solve and then add 2. What do you mean add 2? What do you mean? Oh, I see what you mean. So yeah, it's the standard error of the mean, the 2 SE above and below. So what I'm doing, guys, I'm sorry. What I'm doing is I am adding 0.6 above the mean and 0.6 below the mean. That's what I'm doing. I'm drawing a line, 0.6 above the mean, 0.6 below the mean, and then for sunny leaves, which is the blue, 0.48 above the mean and then 0.48 below the mean. That's what I'm doing, okay? Wait, does it have to be exact on our slide? No, it doesn't have to be exact, because let's talk about our conclusions, okay? Because what can you conclude? Is there a statistical difference between shady and sunny leaves? When I look at this, is there a statistical difference between shady and sunny? Yes, because those bars are not crossing over. Okay, it's not crossing over. Because the 2 SE error bars, or it's called 95 confidence interval, the line, do not overlap between sunny and shady, the shady means. Okay, that mean 7.43 minus 0.6. Okay, this is so confusing. But what this is comparing is like the lowest part and the highest part of the bars, they're not overlapping. They're not overlapping. This strongly suggests the two populations are indeed statistically significantly different from one another. Okay? If the error bars did overlap between the groups, you do not claim a statistical significant difference. Okay? Some of you guys are asking me, Mission, what do you mean by overlap? Okay, here we go. Let me just show you, okay? Good questions, you guys. So, do you guys see these bars right here? These bars are overlapping, okay? See the top right here? The top of the 2 SEM. So let's say this is 2 SEM. And then this is 2 SEM minus, okay? They're overlapped. There's an overlap. You know what that means? Is that, remember this question about the medication? That means that this medication, there's no statistical difference. There's no statistical difference, significant difference, we should say significant difference between these two groups, between these two sets of data. Before medication, after medication, there's no statistical difference. So you know that company that we talked about at the beginning of this Google slide? The company that claimed that their serum can improve red blood cell count after 30-day use. If scientists and AP Bio students saw this graph, they'd be like, the lines overlap. The lines overlap. So therefore, there's no statistical difference between you. They cannot make this claim that this medication works. They cannot make this claim that the medication works because the data overlaps. You might have people here, you might have people before medication, and then after medication, there's no improvement. Okay? That's what it means. So, oh, you guys are funny. So going back to this, there is statistical difference between these two groups. Okay? There is statistical because you guys, you can see here that the lines, there's no overlapping in the 2 SEM bars. Between the 2 SEM bars, there is no overlap, right? There's no overlap. Okay, that's a blue. There's no overlap. So there is statistical difference. So you guys, I want to ask you, like, I can see a question like this being asked on the AP exam. So you have a group of plants that have a mean leaf size of, like, this is the leaf size in the shady versus the sunny. What's going on here? Why is there statistical difference? Why would the leaf size be bigger in plants that are growing in the shade versus plants that are living in the sun? You have to apply your knowledge of photosynthesis to this data, to this graph, okay? And it makes sense that plants that are going in the shade, they're trying to capture more sunlight. So there's going to be more surface area. They're going to be bigger to capture more sunlight, okay? So Tony Stark says, no overlap, statistical difference. Overlap, not statistically different. Correct, okay? So somebody says, Miss Shim is the best teacher, prove me wrong, says, less surface area for transpiration. Oh, that's true too. You know what, you guys, I was just thinking about photosynthesis, but Miss Shim is the best teacher, prove me wrong, says, brings up transpiration. So like if the plants are in the sunny side of the building, they're going to have more harsh sunlight hitting the leaves, greater rate of transpiration, meaning greater water loss, which could be harmful to the plant overall. So they're going to want to have a small leaf size. So somebody asked, people will see this. Somebody asked me, they would give the graph with the lines for us, right? Would we have to figure out the lines ourselves? No, you would be given the lines, most likely. Okay, so again, if the lines do not overlap, there is a statistical difference between the two sets of data. But if they do overlap, there's no statistical difference between the two groups. This pharmaceutical company, remember at the beginning of our Google Doc here, Google slide, I was like, hey, would you guys believe the company? Remember I said that? Would you guys believe this company? And most of you guys said, yeah, I would believe it. I would believe it. This bar looks so much taller than the other bar. But then when they add the 2 SE bars, you're like, no, there is no statistical difference. So actually, guys, when you look at journals, when you look at scientific journals, they always have the 2 SE bars there. They always have the 2 SE bars there. If they only have regular bars, that's not a legitimate data analysis, okay? That's not legitimate. Okay, so let me see here. Demonstrate. So you need to be able to demonstrate 4 quantitative skills: graphing, data analysis, hypothesis testing, mathematical modeling. So let's talk about graphing. You might see bar graphs, and this is the 2 SE, but you would most likely be given, sorry, this is 1 SE. Most likely you'll be given 2 SE, okay? And then scatter plot. Okay, this is a type of scatter plot graph where you see the data points scattered and then you have a straight line. Okay, that straight line is the best fit for the trend in the data. Okay, and then I really want to get to sample problems. Box and whisker plots. You might see this. I've never seen this on an AP exam, which is kind of interesting. But basically a box and whisker plot has a line that shows you the highest data point, and then this line at the bottom is the lowest value in the data set. And then you have this box, okay? This box represents like the 50%, the upper and lower quartile, and then that line inside the box represents the median or the middle of the data set. I am not a reader. One of these days I want to, but I would have to sacrifice a summer for it, and so I would love to be a reader. This is a histogram. A histogram is a bar graph, but it includes numbers or quantitative variables at the x-axis versus, remember this one. This one just shows you the 2 groups, the 2 experimental groups, shady versus sunny, whereas a histogram has like numerical value with along the x-axis and the bars. Okay, I'm just kind of introducing you guys to like the different types of graphs that you might see. Okay, so this is a bar graph versus a histogram. Okay, we'll skip this because you don't need to graph on the AP exam. Okay, so we'll ask that. Okay, so here's a sample problem. Oh, sorry, I skipped. And you know what? I will post the answers to these. You know, if we don't finish on time, what we'll do is I'll just like give you guys the answers and then you can circle it on your Google slide. Okay? Document. What most likely causes the trends in oxygen concentration shown in the graph above? Okay, so this is something that you could see on the AP exam or the quiz tomorrow, sample multiple choice questions using graphing. So if you look here, you can see the time of the day on the x-axis and then on the y-axis, the oxygen concentration. So as the day goes on, the amount of oxygen concentration in the water, so it's very important to also look at the title. You guys, I know it's really easy to just kind of skip the title, but the title gives us a lot of clues as to what this graph is about. Oxygen concentration in the water of a lake. So oxygen concentration goes up in the mornings, and then oxygen concentration goes down at night in the lake. So what mostly causes the trend? It's going up and down. A, the water becomes colder at night and thus holds more oxygen. Have you guys ever like heard that in this class? No. Respiration in most organisms increases at night. Is that true? Do you guys respire more at night? No. C, more organisms are respiring at night than during the day. That's C. You know what? B and C are like the same. It's the same answer, but just worded the same. And then Mick says D, and D is the correct answer. Photosynthesis produces more oxygen than is consumed by respiration during the day. And that makes sense, right? Because light is necessary for photosynthesis. One of the byproducts of photosynthesis is oxygen. So during the day when there's light, oxygen is going to increase. And then there's not a whole lot of photosynthesis at night, so it's going to be going down. But then there's less more photosynthesis happening in the day than respiration. Somebody asked me, is this a sample problem from a past AP exam? It is. This is a question from a previous AP exam. So the answer for that one was D. Let's move on. A group of mice was released into a large field with, I guess we should read the title first, Misham. Fur color distribution immediately after release. Fur color distribution after 20 years. A group of mice was released into a large field to which no other mice had access. Immediately after the release, a representative sample of the mice was captured, and the fur color of each individual in the sample was observed and recorded. The mice were then returned to a field. After 20 years, another representative sample of the mice was captured, and the fur color of each individual in the sample was again recorded. Which of the following best explains a change in the frequency distribution of fur color phenotypes in the mouse population as shown in the figures above? Okay, so A, the allele for gray fur color is unstable, and over 20 years, most of those alleles mutated to become alleles for black fur. Well, first of all, this, allele unstable, like this to me is like nonsense, right? I mean, we haven't learned anything about a allele being unstable and turning into black. That's just nonsense, right? B. Okay, the field was composed primarily of light-colored soil, little vegetation, affording gray mice protection from predators. Yeah, I mean, over time, the amount of gray color increases because maybe they have an advantage, right? An adaptive advantage over brown and black. So the answer actually is B. Let's look at the other one, sexual selection. Nope. The gray mouse is hard to capture. No. Okay. By the way, what type of natural selection is this? Is this stabilizing selection? Is this directional selection? Is this diversifying or disruptive? Good. It's just directional. It's directional. You guys, a long time ago, there were hardly any graphs on the AP exam. It was all about memorizing facts, but now it's all about looking at graphs and applying your knowledge, okay? You need to leave. Okay, that's okay. I'm recording this, all right? Bye. Okay. Yeah, if you need to leave, you can go. This was a question that was asked on the AP exam a few years ago, okay? So this is a graph showing species, oh, look at this. It says plus or minus 2 SEX. That's what those bars mean, okay? Now you guys know what those bars mean, okay? Mean nitrogen influx mean coming in, as a function of species present. N is neither salmon nor bear present. B represents only bears. S only salmon present. And SB, both salmon and bears present. So when I look at this graph, can you guys tell me, let me know in the chat box, like, which set of data is significantly different? Which one is like significantly different? Just by, like, yeah, okay. Yeah, SB, do you guys see that? SB is significantly different from the rest. All the other ones, they're all overlapping. See how they all overlap? Oh wait, actually, you know what? There's no overlap between these two. Sorry, I almost messed up there. Between these two, there's no overlap. But between, this one and this one and this one, there's no significant difference, okay? So right off the bat, we can see SB, which is salmon, both salmon and bears present. That's significantly different. So let's go ahead and read this really quick. Pacific salmon and black bears have often been cited as examples of keystone species. Pacific salmon spawn in freshwater streams but spend most of their lives at sea. When mature salmon return to freshwater streams to spawn, they are preyed upon by bears and other predators. When salmon migrate from their marine habitat to freshwater streams, they bring nitrogen and other marine-derived nutrients that subsequently remain in the areas surrounding the streams, a process called nitrogen influx. So basically what this is saying, when the salmon come into the streams, they're bringing with them something called nitrogen influx. They're bringing in nitrogen, which is a nutrient, okay? because bears hunt them, right? And so they're bringing in nitrogen. Okay, and then I already read that. What's going on? Did I skip something? Oh, okay. Okay. In an investigation, the relationship between black bears and salmon and influx of marine nitrogen into the area around a southwestern Alaskan stream was studied. By the way, this was a question on the AP exam, and so students are like reading and reading and reading. Okay, lots of reading. The investigators established several test plots of the same size along the stream with the following species composition: no salmon or black bears, and bears but not salmon, salmon but not bears, and a plot where salmon and bears interact. Nitrogen influx in the different sampling areas was measured as a means of assessing the impact of the different species on the health of the ecosystem. The data are plotted in figure 1. So the y-axis, okay, I want to ask you guys, what is the dependent variable? What is the dependent variable according to this graph? Good, nitrogen influx. Dependent variable is always on the y. The independent variable is what's being manipulated or studied. It's the amount of nitrogen in the area where there's no bears and salmon, where there's only bears, and where there's salmon, and where there's both salmon and bears. Okay? And so here's our very first question. Excuse me. Oh, that was very rude. I just kind of coughed right into the mic. Which of the following statements is best supported by the data? A, black bears are a keystone species. Can you come to that conclusion just based on this graph? They're a keystone species. I mean, black bears, they bring in this amount of nitrogen, but it's the same amount of nitrogen without bears and without salmon, right? Because there's no statistical difference. Do you guys see that you cannot choose answer A because there's no statistical difference between bears and no bears, okay? B, salmon are a keystone species. Well, you know what, guys? When I compare salmon with no salmon, there's no statistical difference. So B is not the answer. C, black bear's urine is a significant source of nitrogen in the environment. Well, that is just like saying A and C, so C is not it. Okay, everybody's saying D. D is the correct answer. The presence of black bears and salmon correlates with a significant increase in nitrogen influx. That's the only one that is truly significantly different, so D is the correct answer. By the way, do you really need to know a lot of AP Bio information to answer these questions? No. Okay, I would say like half of the multiple choice questions, you don't need to know a lot of AP Bio, but you are going to be so ready for the AP exam. You are going to have background knowledge. That's why it's very important. You have a lot of background knowledge and you're ready to answer these questions. So even though you could, that's why you could have a really crappy teacher and not know a lot of AP Bio and still do really well because, 50% of the multiple choice is based on common sense, okay? Yeah. Okay, number 7. But you know what? Like keystone species. You do need to know what that is to answer, to say, oh, they're not that important, okay? But again, like these type of questions is just practice, okay? Which of the following most likely describes how the interaction between bears and salmon influences nitrogen dynamics in the environment? A, when bears consume salmon, they leave parts of the carcasses on the ground, which decompose, releasing nitrogen into the environment. That's very much a possibility because when salmon and bears are together, the bear eats salmon, leaves behind the carcasses, and then the nitrogen influx is significantly different, right? But let's take a look at answer choice B. When salmon swim upstream to spawn, bears migrate to the rivers and deposit nitrogen from the forest with their excrement. That's a possibility. Basically, what it's saying is that the bears follow the salmon, and then their poop is the thing that... But then you can argue that wherever the bears are would bring in a nitrogen influx, a significant nitrogen influx, but then again, it's not statistically significant when there's no bears around, okay? C, bears are a reservoir for nitrogen because they are the top No. D, bears urinate in rivers and streams, which provides nitrogen for the salmon. No, you guys, so the answer is A. A makes the most sense. Okay, A makes the most sense. Okay, next. You know, I feel so weird. You guys have all the, you know, your actual names are not in the chat box, so it makes me feel a little bit sad. Like, it's not, like, as personal, but that's okay. Okay, 8. Which of the following pieces of additional data would help further investigate the relationship between bears, salmon, and influx of nitrogen into the local environment? So, like, in other words, which of these, you know, parts of the experiment would help us better understand what this is studying? You guys are... what's 7 again? 7, the answer for 7 was A. It was A. Okay. All right, let's look at answer choice A. And you guys, I mean, I say 4:30, but I can stay a little bit longer. A, the experimenter should remove the remains of salmon carcasses immediately after the salmon are eaten by the bears and determine the nitrogen content of the carcasses. Okay, A is a possibility because we did say that when the bears eat the salmon and they leave behind the carcasses, that's contributing to the nitrogen influx. But if we remove the salmon carcasses right away, then we can study just how much nitrogen is in the carcasses, right? Good question. Somebody asked me, do we need to write these answers somewhere? Yeah, in the Google Doc, you're supposed to like circle the answers, okay? So you're like circling the answers. And if you didn't know that, that's okay. Maybe you can write the answers right now, okay? So answer for 6 was D. 7 was A, right? 8, that's what we're doing right now. So we said A is a possibility. Let's get B. The experimenter should increase the number of bears in the area and measure the amount of nitrogen. That's just too much. Why would you bring in bears, right? Yes, you can draw. So go to the line, or you could do shapes. You could do shapes, you could do circle, okay? But if you go to line and scribble, do you see my shared screen? So go to line and then scribble, okay? Or you could do a circle shape. Okay. Let's look at C. The experimenter should set up... Okay, I'm going to cross it out. The experimenter should set up a net to catch salmon before they enter the area. But they already did that. Didn't they already do that? Here and here. There's no salmon in the N and the B, right? So they already did that. They already studied where in an area where there's no salmon, okay? D, the experimenter should clear out the trees from the area and determine how much nitrogen remains. That's silly, right? So A is the correct answer. Yeah, you're basically answering these questions and turning them in, okay? 9, if a dam is built downstream and prevents salmon migration to the test sites, which of the following most accurately predicts the impact on nitrogen influx? This is basically saying, what if there's no salmon coming into the area? A, nitrogen influx will increase because the bears will no longer store nitrogen from the salmon? Does that make sense? No. Nitrogen influx will decrease because there will be less bear-salmon interaction? B is the correct answer, everybody. B is the correct answer. Okay? We don't have time to go over all the answer choices. But what I'm trying to emphasize is that with the exam, everyone, it's all about practicing and exposing these type of questions, being exposed to these type of questions. Okay? Oh, man. I'm looking at this question. Did I put it on your quiz tomorrow? I'll have to double check. I might have to edit the quiz. I might have put this question on the... Did I do that? Okay. So you guys, basically, really quickly, I don't think I want to read through this whole thing. In an experiment that students did, they were basically investigating how light affects the carbon cycle in an aquatic ecosystem. So they have like a plant growing in water with light, a plant growing in water in the dark. You have a goldfish in light, you have a goldfish in the dark, and then you just have two beakers, one in the light and one in the dark. Okay? So, like, which one do you guys think which group is the control group? It's going to be 5 and 6. 5 and 6 are the control groups, and then groups 1, 2, 3, and 4. Those are the experimental groups. And what the student did was they measured the pH. They measured the pH, and then notice how they add the standard error of the mean. And if you were to graph that, which you don't have to do, you would multiply that by 2. Okay? So you guys, what's the dependent variable? What's the dependent variable? What's being measured? If you were to graph this, what would you be graphing? Like, what would be on the y-axis? pH. Good, pH. Yeah. How do we know if we answered some of the questions correctly? I missed a few of them. Sorry. You know what? I think that when I grade these, I'm just grading them to see if you did it. Okay, so... But you should try to get the correct answer, okay? Oh, also, I'm recording this, so maybe you can go over I am recording this, so you can go over the answers, find the answers there, okay? So 33, which of the following graphs is most appropriate representation of the experimental results documented in the table? So this is the data results, pH, groups. So which way of representation is the best? Is it A with the pie graph? Is it a bar graph? Is it a line graph? Or is it this crazy line scatter plot? Yes, B, you guys, B is the correct answer. So B is the correct answer. Notice how they added the standard error of the mean. Yeah, I would say C is the second best. You know what? On the AP exam, like if you were to graph, which you don't have to do, both B and C would be acceptable. B and C would be acceptable. However, why are you connecting those groups, right? You don't need to connect the groups because usually when you connect dots, that's a whole process that's happening. So you don't, why are you connecting the dots? You don't need to connect the dots. Okay, let's see here. Oh, you guys, this is important. You don't have any more grid-ins, but they always ask, every year they ask, how do you find the rate of a reaction? Okay? And the rate of the reaction is the slope of the line, okay? And in your math class, you know how you find the slope, right? Isn't it just, isn't this the equation? Isn't it just like y2 minus y1 over x2 minus x1? That's how you find the slope of something, or the rate of a reaction. So I put this in here because you will see, or possibly see, a rate of reaction question on the quiz and then on the AP exam. Calculate the average rate of increase in oxygen consumption for animals acclimated to 5 degrees Celsius. Give the answer, and this is what they want you to know, okay? So 5 degrees Celsius, that would be this line with the open circles. So we're looking at this. Temperature increase from 10 to 30. So from 10 to 30. So what you need to do is y2, so this is y2, which is like 180 minus y1, which is over here, 80, divided by x2, 30, minus 10. So this is how you would solve this problem, like that. And what is that, like 5 or something? Am I wrong or right? 5? Is it 5? I'm so bad at math, okay? But that would be the rate of reaction, okay? Thanks, guys. All right, so that's the rate of reaction, 5. It would be like 5 milliliters of oxygen per gram per H per degree Celsius. Okay? All right, and then maybe I should just give you guys answers, huh? Because it's already time to go. Oh, and you know what? I'm sorry, but I didn't. Should I just pull up the PowerPoint? The PowerPoint has all of the answers, so maybe I should. instead of going over it with you guys. And then if you guys, I could just put it up in the update. That's true. Hold on one second. Let me open up my desktop, AP Bio. No, that's not it. Now you guys are seeing my folders in the shared screen. Oops, no, it's here. But you're not in the Zoom. Yeah, you can't see it in the Zoom, but you can see it in the shared screen whenever. Okay, so I just don't want you guys to have to look at. Oh, wait, hold on. Okay, so for the next question, go to the Zoom. The answer is A. Okay, so for the next question, the answer is A. So the answer is A. That way we can practice the problems and just check to make sure that we got the right answers. Oh, so you guys want me to just put it in the update and not in this shared, in this screen record? Is that what you guys would prefer? To not have it in the screen record? Okay. Somebody says yes, please. Or either way should work. I think it's just easier to just go over, just to give you the answers right now. And then the next answer is B. Okay, so the answer is B. Sorry, guys. So the answer is B. So you'll circle B. Dude, I am recognizing some of these questions that... okay. And then the next one... Oh, the next one is not on here. Okay. I'll just have to double check then. Then I'll just have to put this in the update in Schoology since we don't have time to like figure it out. Oh, that's inconvenient. That's okay. Okay, so then for question, this question 52, 53, 54, 55, 56, I'll have to put that in an update in 56 and 57. Okay, slide 50. Somebody's asking what was the answer for slide... This one was B, right? Slide 50, the answer was B. Okay, I will... Okay, this is what I'll do. I'm going to put... Oh, wait. Somebody said it was A? Let me just double check. Oh, no. It was A. It was A. I'm sorry. It was A. Oh, okay. You guys, I'm sorry. A. It was this that was B. So 51 was B, yeah, and 50 was A. I apologize. Okay, so in an update, I'm going to link to the screen record, and then in the update, I will include the answers to the rest of the slide. But then that's not going to make you guys try to find the right answer. Will you guys really try to find the right answer on your own and then check your answers? Will you guys do that? Tony says yes. Okay. Yes, so how do you study? Just do the problems. Okay. Yes, we are academically responsible. Okay. All right, I'm gonna stop the screen record then.